This is Lab 7, Introduction of Enzymes, by Navneet Plaha and Bobby St. Pierre. What are enzymes? Enzymes are usually proteins that act as catalysts to speed up different reactions in the cell. Note that they can sometimes be like ribozymes. But anyways, a catalyst is an agent that speeds up the rate of a chemical reaction without being changed or consumed in the process. Enzyme structures. Enzymes are usually large proteins and they have an active site. The active site is an area that an enzyme binds to a substrate. And over here in the picture below, you can see like a very realistic model on the left side and of the active site and then the substrate binding to it, and this is the very large enzyme. But then there's like a schematic model, very much more simple, and the yellow blob is basically the enzyme. And this orange structure is the substrate binding to the enzyme at the active site. The active site. As stated previously, the active site is an area on an enzyme that binds to a substrate. And a substrate is a molecule that molecules that participate in chemical reactions. An enzyme is specific to its substrate. It cannot bind with anything else that does not have that shape. It's like how only certain keys can fit into a lock, and the key could be an analogy to the substrate, and a lock can be an analogy to the active site. How does an enzyme work? Well, first, a substrate binds to the active site of an enzyme, then the enzyme speeds up the rate of the chemical reaction, and then finally the chemical reaction is finished and the products leave the enzyme. And you can see in the picture below, the substrate comes to the enzyme connecting at the active site, it gets catalyzed, and then the products leave the enzyme. Induced fit theory. This theory was proposed by American chemist Daniel Koshland. It states that sometimes when an enzyme binds to a substrate, it changes shape to hold the substrates more tightly. Once the substrate is converted into products, the enzyme releases them and then goes back to its original shape. Activation energy. In chemical reactions, each molecule has an electron cloud that causes repulsion. Activation energy is the initial amount of energy needed in a chemical reaction to get the molecules close enough to have a rearrangement of bonds. Activation energy is used to get to a transition state, and a transition state is the most unstable state of any molecule because it is when the molecule's bonds have been stretched to their limits. Enzymes' roles in activation energy. Activation energy in certain chemical reactions can be very high and would take too long to take place on their own. Enzymes lower the activation energy so that less of it is needed for the chemical reaction to take place. Without enzymes, chemical reactions would go a lot slower in cells. And for example, the enzyme catalase speeds up the breakdown of hydrogen peroxide to water and oxygen 10 to the 15th power times faster than without the enzyme. And in the picture below, it shows how a certain chemical reaction will need a certain amount of activation energy, and you can see in the red line that it's very high without the enzyme, but the blue line showing it with the enzyme shows that the enzyme lowers the activation energy to a much lower state, so that not as much of it is needed for the products to be created. Inhibitors. Competitive inhibitors are molecules that bind to the active site of an enzyme to prevent a substrate from binding to it. They can bind with the active site because they have structures similar to the substrate. Non-competitive inhibitors are molecules that bind to the allosteric site of the enzyme to change the conformation of the enzyme and its active site, therefore inhibiting the ability of a substrate binding to the active site. An allosteric site is a location outside of the active site. And as you can see to the picture on the lower left, there is a reaction on the top that's just very normal. A substrate goes into an enzyme, you're connecting through the active site, then it gets catalyzed and it releases its products. But below, it shows that the active site has a competitive inhibitor placed into it, and that prevents the substrate from ever actually reaching the active site, so it can never be catalyzed. Then, on the right with the white picture, you can see on the top there is a normal reaction, a substrate binds to the enzyme through the active site, gets catalyzed, and then gets released as products. But then in the bottom, you see that there's an allosteric site as well. And a non-competitive inhibitor attaches into the allosteric site. It changes the conformation of the active site, changing its shape. So then a substrate molecule cannot actually fit into the active site, so then the chemical reaction can't really take place. How is enzyme activity regulated? Enzyme activity can be regulated by competitive and non-competitive inhibitors and deviations in its environment. Environment for optimal enzyme activity. The environment an enzyme is very important to its function. Any major deviations in its environment and the enzyme activity can be seriously impaired. Human enzymes function at human body temperature, which is 98.6 degrees Fahrenheit. The pH an enzyme works in depends on the enzyme. While some prefer more acidic environments, others might prefer more basic environments. Not very basic, but just slightly. And ionic conditions can also affect the enzyme's ability to function properly. Enzyme activity and substrate concentration. If there are more enzymes for a specific substrate than there are substrates, then if you add more substrates, the rate of reaction will increase. However, if there is more substrate than enzyme, no matter how much more substrate you place in the cell, the rate of reaction cannot increase, and that is known as the enzyme saturation point. And in the picture shown below, you can see that there's a low amount of substrate and not all the enzymes are being used. But then when you place more and more substrate into it, more enzymes are being used, so then the rate of reaction is increasing. But then, when there is a high amount, like a very large amount of substrate in the cell, all of the enzymes are already being used, and no matter how much more substrate you put into it, the rate of reaction cannot increase. It's already at its maximum. And the little blue diamonds are the substrates, and the red blobs, I guess, are enzymes. Allosteric regulation. Allosteric regulation is a way for non-competitive inhibition to control whether or not an enzyme is catalyzing a substrate or not. Allosteric regulation can be used when the cell has too much of a certain substrate being catalyzed, because enzymes, when they catalyze certain substrates, it takes up energy, and that's just wasteful when it's not needed. And you can see in the picture in the bottom right that there is a protein, and at its allosteric site there is a regulatory molecule not attached to it, and the active site, the substrate is attaching to it, and then getting catalyzed and turned into products. But when the regulatory molecule does attach to the allosteric site, the active site changes shape, and the substrate cannot fit into it anymore. Thank you for watching.